Good morning, students. Today we discussed about senescence in plants and programmed cell death. Every autumn, people who live in temperate regions can enjoy the beautiful color changes that precede the loss of leaves from deciduous trees. So profound are these color changes in forest of such trees that they are apparent even to observe in orbiting spacecraft. These leaf changes color because shorter day length and color temperature trigger development process that are collectively known as senescence. Although it leads to the death of targeted tissues, senescence is distinct from necrosis, whereas necrosis is death brought about by physical damage, positions, or other external injury. Senescence is an energy dependent development process that is controlled by the interaction of environmental factors with intrinsic genetically regulated programs. In this last section, we describe various types of senescence in more detail and we present evidence that senescence represents a programmed and adaptive aspect of development. Plants exhibit various types of senescence. Senescence occur in a variety of organs and in response to many different cues. Many annual plants, including major crop plants such as wheat, maize, and soybean, abruptly yellow and die following fruit production, even under optimal growth conditions. Senescence of the entire plant after a single reproductive cycle is called monocarpic senescence. Other types of senescence include the following: senescence of aerial shoots in herbaceous perennials, seasonal leaf senescence as in deciduous trees, sequential leaf senescence in which leaves die when they reach a certain age, senescence ripening of fleshy and dry fruits, of storage cotyledons and floral organs, senescence of specialized cell types like trichomes, tracheids, and vessel elements. The triggers for the various types of senescence are different. They can be internal, such as reproductive processes in monocarpic senescence, or external, such as day length and temperature in the autumn leaf senescence of deciduous trees. Regardless of the initial stimulus, the different senescence patterns may share common internal programs in which a regulatory senescence gene initiates a cascade of secondary gene expression that eventually brings about senescence. Because of its effect on agriculture and storage of agricultural products, the regulation of plant senescence has received considerable attention in the past years. Here, the model plant Arabidopsis has served as an important model species. The genetic aspects of senescence regulation have been investigated using two complementary approaches. First is the study of genes that are upregulated during or prior to the onset of senescence, and the second is the analysis of genes that cause an altered senescence syndrome when muted or ecologically expressed. The expression of many genes changed dramatically upon The initiation of senescence. Not surprisingly, the expression of photosynthesis related genes decreases, but at the same time, the expression of thousands of other genes increases. Such genes, called senescence associated genes, include regulatory genes such as transcription factors and genes involved in degradative process. SAG include genes that encode hydrolytic enzymes such as protease, ribonuclease, and lipase, as well as enzymes involved in the biosynthesis of ethylene, such as 1-aminocyclopropane, 1-carboxylic acid synthase, and ACC oxidase. Other SAG have secondary functions in senescence. These genes encode enzymes involved in the conversion or remobilization of breakdown products such as glutamine synthase, which catalyze the conversion of ammonium into glutamine. Pectins play important roles in cell wall breakdown during leaf abscission and fruit ripening. Several key findings illustrate the complexity of senescence regulation. Environmental cues such as changing day length and temperature as a result of changing seasons affect senescence in deciduous trees, while stress, including pathogen and temperature stress, induce leaf senescence in many plant species. Hormones control senescence. Ethylene has long been known to induce senescence, while cytokinin is a potential inhibitor. Mutants that have a defect in ethylene perception generally show delayed senescence. Likewise, plants with increased cytokinin sensitivity show delayed senescence. In addition, most other plants' hormones affect senescence to some extent. Next is oxidative stress can damage proteins and cell structures. The accumulation of cellular damage has been proposed to cause senescence in a number of species. However, plants have large capacity for detoxifying reactive oxygen species, so oxidative stress more likely function as a signal than a direct cause of deterioration and subsequent senescence. Metabolic status regulates leaf senescence. Sugars accumulate during senescence, and the sugar sensor hexokinase contribute to this process. Macromolecule degradation can affect senescence in multiple ways. Arabidopsis mutants with a defective ubiquinin dependent proteolysis pathway show delayed senescence. This finding suggests that senescence may be induced when factors that inhibit the senescence syndrome have been degraded by targeted proteolysis. The senescence syndrome itself involves massive macromolecular degradation after senescence has been initiated. And next is intrinsic development factors. IDF. It prevents leaf senescence during early developmental stages and induce senescence during late development. Thus, senescence depends on age related changes even in environmentally adverse conditions or after treatment with the senescence inducing hormone ethylene, young leaves will not senescence. Conversely, no mutants have been isolated and No environmental conditions have been found in which plants stay functionally green forever. It's completed. Thank you, students.